Thanks, Jessica. Leah?
Thank you. I appreciate the opportunity to be a part of this.
Could you sit up nearer the microphone? Thank you.
I do appreciate being invited to be a part of this workshop and hear the different perspectives and thoughts on this sort of emerging topic. I am representing the Industry Pharmacogenomics Working Group, and by industry, that refers to the pharmaceutical industry, and therefore my comments and perspective focuses largely on clinical trial sample test results and mostly of a genomics nature. So there are many categories of research results generated from clinical trial biospecimens, including inclusion-exclusion criteria, endpoints, safety monitoring, pharmacokinetics, including genotyping for drugs, ADME enzymes, which control the absorption, distribution, metabolism, and excretion of the drugs being tested in the trial, and also for exploratory research, which may be conducted either during the trial or up to several years after the trial is completed, at which time there may no longer be contact between the participant and the investigator at the study site. The results of clinically relevant CLIA-certified laboratory tests done for safety monitoring are returned to the investigator at the study site if the results indicate a potential medical concern for the subjects. And then the site would follow its procedures for sharing and acting on that information. The investigator may recommend confirmatory testing outside of the clinical trial. They may notify the subject's primary care physician, or they may take other action. Of note, the clinical trial sponsor, the pharmaceutical company, does not return results directly to the subject, and in fact, we do not know the subject's identities. Aside from some safety labs, very few tests run on clinical trial samples are both CLIA certified and yield clinically relevant results. In these uncommon cases, the tendency is to return results without interpretation, again, to the investigative site, which then would follow its procedures for sharing and acting on the information. However, sometimes this is not feasible, for example, if tests are performed after the trial is over. Results that are not clinically relevant and/or from CLIA-certified tests are typically not returned to subjects, and most test results performed on clinical trial specimens are of this type. Many of these tests, for example, tests to understand the biology of drug administration and action, they are exploratory and have no established interpretation and may only be useful when analyzed in aggregate. So these results are for research purposes only, and it would not be appropriate to base treatment decisions on them. As has been referred to before, a suitable method and associated infrastructure must be established to return results responsibly, and the resources involved can be considerable. As next-generation sequencing and whole exome sequencing are used in scientific research, these platforms will create particular challenges for returning results due to the vast amounts of data they generate. And in addition to other infrastructure elements, in the case of genetic data, access to counseling by a trained professional with the requisite expertise may be appropriate. As was mentioned earlier, pre-analytical variables can affect the accuracy of the test. Any results from samples known or suspected to have been collected, shipped, processed, stored, etc., under adverse conditions would be questioned and annotated with that caveat when used for internal decision making, and they would not be returned to the subject in any case. In the pharmaceutical R&D setting, flexibility and speed are critical in developing new assays for use in timely decision making in our drug programs. So while validation to CLIA standards provides a measure of quality, it's not always practical or even necessary to ensure the results are fit for purpose. And mandating such validation could indeed impede innovation and/or introduce delays to the drug R&D process. So with that, I'll conclude, and thank you again.